It's a great pleasure for me to introduce you all to our cyber lecturer today, Dr. Dolores Hambardzumyan. Dolores joined us in the fall of 2019 from Emory University. She was here just for a few months before we faced last spring's lockdown, but despite that has really hit the ground running. And we're so thrilled to have Dolores on our faculty. Her focus is on the biology of brain tumors, and it goes along with one of our strategic initiatives within the Friedman Brain Institute with our partners in the Department of Neurology and Department of Neurosurgery in the Tisch Cancer Institute to build an interdisciplinary team focused on the biology and treatment of brain tumors. So we're delighted to have Dolores at Mount Sinai and to speak with us today. So Dolores, take it away.
Thank you so much, and thank you, everyone. And I am very happy to present today. Okay, let me see. Okay, perfect. And okay, and today I'm going to talk about myeloid cells, cells of your innate immune system in high-grade gliomas. Before I start, I always like to acknowledge my lab members. This is my small lab in Mount Sinai, and I want to especially thank my former lab members, two outstanding graduate students whose work I'm going to present today, Cameron Herting and James Roth, that are now postdoctoral fellows in a pretty prominent labs. And I want to thank, of course, Z, who's been instrumental driving most of the work today I'm going to present, and he's a faculty here at Oncological Sciences at Mount Sinai. And I want to thank all my collaborators at Emory University, at Mount Sinai, and my main collaborator also that I work with the pediatric brain tumors, Soren Becker at Northwestern. So, and I want to thank funding agencies without whose support this work wouldn't be possible. So today I'm going to mainly talk about glioblastoma, which is the most common and aggressive primary brain tumor that affects around 17,000 individuals annually in the United States. The median survival with maximal surgical resection and radiation therapy is around 12.1 months. As you can see here that you have the red line, and the only addition of temozolomide, which is alkylating agent, to this regimen extends the survival where it currently stands at 14.6 months. And, you know, it's been 50 years, we really didn't do much besides just shifting the survival for 2.5 months. It's extremely difficult tumor to work with, and many reasons why this tumor is resistant to therapy, but one essential factor is the heterogeneity of these tumors within neoplastic cells, within tumor cells, consisting of both inter and intratumoral heterogeneity. And when we refer to inter and intratumoral heterogeneity, we always think about tumor cells, and I hope by my talk today, I will convince you that this heterogeneity actually translates into heterogeneity, both inter and intratumoral in tumor microenvironment that I'm going to talk about. So over 15 years ago, probably, because this tumor is very heterogeneous, that's how they got the name originally, glioblastoma multiforme, although we don't use it anymore. But 15 years ago, the Cancer Genome Atlas Initiative, based on RNA expression profile, cataloged these tumors into preneural, classical, and mesenchymal subgroups. These subgroups are not mutually exclusive. On contrary, you can have mix of the cells within the preneural tumor. You will have cells with classical and mesenchymal signature. They are just pretty much based on the dominant number of cells that you have. For example, in a preneural tumor, majority of your cells will have the preneural expression signature, while in classical, and mesenchymal. But it's easy, those subtypes are very plastic. You can hit with treatment with anti-angiogenic therapy or radiation therapy, the preneural tumor, and it easily will convert into mesenchymal subtype. Not ideal division, but at least something. And today with single cell sequencing data, people are dividing it even in more subtypes, but nevertheless, all of them point to one thing: these tumors are very heterogeneous. And in 2016, WHO divided these tumors into IDH1 mutant and IDH1 wild type. And the reason they did this, because the IDH1 mutant is actually a defined subset of patients that have much better survival compared to IDH1 wild type. So it's fair to say we have this small subset, around 6% of tumors that live and do much better than IDH1 wild type tumors. And this IDH1 wild type tumors, further we can divide based on RNA expression into preneural, classical, and mesenchymal. So those are based on RNA expression. Modeling RNA expression, it's very difficult. But if you add the genetic heat map into the subtypes so that we can use that information for modeling, you can see from just first look that mutational burden for glioblastoma is very low. But if you have a closer look, you can see some common tumor suppressors like P53, INK4AR, PTEN. Those are universally lost or distributed in all the subtypes. But if you look at the GFR signaling, PDGF signaling, or NF1 loss, you can see that although mutually not exclusive, they cluster in certain subtypes, meaning like majority of PDGF amplified tumor cluster in preneural subgroup, while EGFR mutants or EGFR amplified in classical. And NF1 is cleaner. The tumors with NF1 loss around 11%, those are more clustered together into mesenchymal signature. The reason I brought this up because this is the driver mutations that we use to model glioblastomas in mice, so we can do some immune microenvironment related studies. Just to touch base, and then we have pediatric high-grade glioma, including glioblastoma in pediatrics, which for a long, long time we thought it's the same like adult, but those are completely different tumors that we learn thanks to RNA sequencing and genomic studies. This tumor share a common mutation with adult, and that's only PDGF receptor amplification. And then you have 50-50 split with the histone mutant tumors that happen specifically in midline and brainstem, and then you have hemispheric tumors. So they are very specific to location, and specific histone mutations such as H2.1, 3.3K27, or G34RV mutation that usually don't happen in adult and very specific to pediatric subset. And those are the genetic alterations exactly captured in this picture that we use to generate the same locations that we capture in humans using mouse models. Now, before I go in and introduce to genetically engineered mouse models that we use, it always makes me smile. The George E.P. Boxes was a British biostatistician board that all models are wrong, but some are useful. But by no means I want to convince that genetically engineered mouse models are the way to go. They are just a tool, and I think for immune microenvironment and tumor cell interaction where species compatibility is essential, I think these models are quite useful. The system that we use to generate tumors in GM models is called ARCAS TVA. ARCAS is a chicken virus. We do not have then, that's the basic structure of the virus. You have LTR, GARPO envelope, and that's where we put any oncogene of interest that we want to overexpress, or we can silence any tumor suppressor that we want by using siRNA approach. So mammalian cells, including mice, they don't have the receptor for ARCAS virus, so they are not infectable by ARCAS viruses. But we have genetically engineered mouse models where the TVA, which is the receptor for ARCAS and necessary for ARCAS virus to get into the host cell and integrate into host genome, is expressed on the nesting promoter. For example, we have multiple of those, but today across my talk, I'm going to talk about nesting because we want to keep the cell of origin consistent. So all the cells in the mouse brain that express nesting will have the TVA receptor, and these cells are the ones that we are infecting with ARCAS viruses. So ARCAS will bind to its receptor, it's going to get into the host cell, nesting positive cell in this case, it's going to integrate into host genome, and mammalian cells have deficiency assembling the virus, so they are not going to produce the virus, and we will achieve only oncogene overexpression that we want, or we will silence tumor suppressor that we want. The model can be used, and we have been using them both for adult brain tumor generation, and that's when you do stereotactic injections and you deliver your ARCAS viruses specifically at the regions of the brain you are interested that are enriched by nesting positive cells, which is in adult brain stem progenitor population. And then you can use the same at P0 to P2 postnatal newborn mice and deliver into brain stem or in a hemisphere, and you can generate hemispheric and brainstem gliomas, the IPGs. So this is just the survival curves of the tumors that we generate using main oncogenic drivers known in a human disease. It's a PDGF, NF1 silencing, or EGFR-V3, which is a human mutation. And we overexpress this mutation in a mouse, and we end up having tumors that resemble glioblastoma and or anaplastic astrocytoma, which is a grade 3, also high-grade gliomas. Murine GBM closely resemble their human counterparts, both in imaging properties. This is MRI, T1 contrast enhancement with gadolinium that gets into the brain. And this is a human, you can see, because brain-blood barrier is compromised and you are able to deliver gadolinium there, otherwise you wouldn't be. And the same thing we see in T1 contrast enhancement, that gadolinium gets into mouse tumors and beautifully enhances in these tumors too. On histology and gross pathology, these tumors captured the key features of glioblastoma, which is the structures called pseudopalisading necrosis. And so you have the necrotic core and you have the rim of beautifully aligned cells, and those are the hypoxic areas and most resistant to therapy. You captured the same structures in murine models, and you have microvascular proliferation in both cases. So this is RNA expression. We did 30 panel RNA in cancer intrinsic signaling pathways, that genes that we did 30 panel qPCR, human and mouse, and we aligned them against each other in a PCA. As you can see, PDGF-driven tumors clustered closer to preneural human, classical and the GFR together, and then F1 and mesenchymal tumors together. So they kind of resemble the heterogeneity we do see in human when it comes to intrinsic cancer cell signaling pathways. So with this, we were kind of a bit confident that these models will be useful to study tumor microenvironment. And by this, I'm going to just jump into tumor microenvironment. So tumor microenvironment is the environment when tumors form in the brain and grow, and they form beautiful interaction with tumor cells. And they also change, although cells in tumor microenvironment are genetically stable, but they are also getting a lot of signals from tumor cells, and they can activate their expression profile. So if we take GBM in mice, we dissociate, which is similar to what we get quantitatively in human, dissect this tumor mass, do mechanical or enzymatic digestion into single cell suspension using various reporter mice, because we don't have surface marker for all the cells in the brain, but we have reporters. In the mouse models, we can do this and do basic FACS analysis. You can see only in a pie chart, only 65% max of this tumor mass that we resected is actually constituted of tumor cells. The rest, 35 to 40%, is tumor cells of tumor microenvironment. And the most abundant cell population in tumor microenvironment in GBM is a tumor-associated macrophages. So today in my talk, I'm going to focus more on intratumor heterogeneity of TAM compartment in glioblastoma. And I will finish my talk telling how important also is to understand intertumoral heterogeneity. So before this, we use the term TAM as but it's consists of mixed populations. But before I introduce how they look like in GBM, I want to bring back to their progeny, where these cells come from. So we have microglia, which comes in from yolk-sac progenitors early during development, and this stays there. The cells are very unique cells of CNS. They can self-renew similarly like stem cells, and they can live pretty long. That's what you have in the brain. In a parallel universe, pretty much, you have bone marrow that produces through the progeny monocytes. And monocytes exit bone marrow. In response to monocytes chemoattractant protein gradient, exit bone marrow, get into circulation. And in circulation, you have two types of monocytes. A subset that it's called inflammatory monocytes, and subset that it's called circulating monocytes. Now, in contrast to microglia, these cells are progenitors. They are not long-lived, so they have half-life probably 12 to 19 hours for inflammatory and a day or two for circulating monocytes. So then die if there is no inflammation. And bone marrow constitutively produces and pumps them into circulation. The picture changes. Now we have a GBM. We have a compromised or leaky vasculature in glioblastoma, which is compromised but not uniformly. So you have areas of leakage where you have areas that are more intact. But you also have brain tumor cells that express high level of monocytes chemoattractant proteins. So similar what happens with the gradient that monocytes use to exit bone marrow get into circulation. So tumor hijacks that mechanism and produces high level of MCPs. Then you have leaky vessels. And these inflammatory monocytes can exit circulation. They get into CNS and quickly, which I'm going to show in a next slide, differentiate into monocytes-derived macrophages. And when they do that, and when you have also microglia in response to tumor activated, they become indistinguishable for microglial cells. But it's important to understand the differences. As I defined, their origin is different, their lifespan is different, and I'll show that their location in tumor is different. So for us, for my lab, the key question was to understand what brings the cells into CNS. Is it the leaky blood vessels? Is it the chemokine gradient, or both? And to do that, we also needed to develop markers and tools to distinguish microglia from these cells to be able to lineage trace these cells. To do that, we took advantage of Christmas mice, and that's Z who names this mice Christmas mice, because we took advantage of CX3, CR1, GFP, and CCR2, RFP, no-gene mice. So those are no-gene. So heterozygote mice will lose one copy of the gene, will be replaced with GFP or RFP reporter, and we will be able to trace these cells. Now, CX3, CR1 in CNS healthy, exclusively expressed by microglial cells, while in circulation, monocytes express both. The intensity, though, varies depending from the stage of differentiation that monocytes has. So you have both express low GFP and high RFP. So using this mouse, we could trace microglia and distinguish them from monocytes. And to do that, we use the mouse. And if we do naive brain, we all know if we enrich with Perkel gradients for microglial cells and plot with, again, CX3, CR1, GFP, and CCR2, one thing we can see for sure that microglia is CCR2 negative and highly positive for CX3, CR1, which is expected. And when we did the same experiment in a tumor-bearing animals, we can see that we have microglia, and then we have large populations, around 85% of macrophages in a tumor are come from bone marrow. You can see they are also distinguished as CD45 high hematopoietic origin versus the microglia that is defined as CD45 low. Similar to what we see in a naive mouse, this is the GFP RFP plot. You can see that microglia is actually GFP positive, CCR2 negative, meaning even in GBM context, microglia doesn't upregulate CCR2 expression. And when you look at the graph for CCR2, you almost see transition stage. You have LY6C high inflammatory monocytes, high for CCR2, low for CX3CR1 expression. And then you see like a curve of differentiation. And if we quantify that, we can see those are newly infiltrated, more predominant red monocytes. Those are macrophages when you have GFP intensity increase and RFP intensity decrease, macrophages that derive from these monocytes. And then you have around 16% of bone marrow derived macrophages that at the end stage of differentiation completely downregulate CCR2 expression. To validate that we are concluding this right, we used multi-parameter FACSes, but to say this is newly infiltrated monocytes, this is monocytes derived macrophage, this is our microglia, and then we have also a rare another myeloid population from circulation coming. And those cells are called neutrophils, but they are much less. Although in the circulation, they are 7 to 10-fold higher than inflammatory monocytes are. But for some reason, they don't come to the tumor as much as monocytes do. So then we said, okay, if that's true, if this is inflammatory monocytes that come, if we use CCR2 RFP mouse, which is basically equals to homozygous loss of CCR2 receptor, then will decrease newly infiltrated monocytes, and it is decreased. The number of monocytes-derived macrophages will decrease too. So it's true, those are CCR2 positive inflammatory monocytes that infiltrate the tumor and differentiate into tissue macrophages. Now, we wanted to understand how early these tumors come, but I want you also remember this. We do decrease their infiltration with CCR2 RFP mouse, but we don't eliminate, they still come. But so to understand how early during tumor development these cells come, we looked at different stages, and this is when you look at the tumor, you are not visible even, and until you have different sizes of the tumors that are very visible. And then you look at RFP, GFP intensity, you can see that GFP is all in the rest of the tumor. I expected microglia is uniformly distributed there. And then you have inside of the tumor, which you can clearly see with DAPI density nuclei, that all the red cells are in the tumor. And we quantified the ratio of red and green cells single, which are microglial cells, and red and green combo is the bone marrow derived cells. And you can see that a ratio doesn't change. So they come to the tumor very early during development. And even when you have a small nodule of the tumor that you can barely fit into 20x magnification under the microscope and look at the red and green distribution, you can see the inside of the tumor, majority of the cells, similar to what we see like FACS, are dominated by CCR2 positive inflammatory monocytes and monocytes-derived macrophages, where in a peritumoral area, it's activated microglial cells. So the location, so the bone marrow-derived cells are inside of the tumor, while microglia is surrounding peritumoral areas of the tumor. So we wanted to, a few years ago, then didn't have yet single-cell sequencing, and we're not doing that with single-cell sequencing. We did RNA sequencing when we isolated naive microglia from the brain. We isolated from circulation inflammatory monocytes, and then did the tumor-associated microglia and tumor-associated macrophages, and did the 6-way comparison, and then narrowed down into tumor microglia versus the tumor macrophages. Not surprisingly, they have 530 genes in common. They are both macrophage lineages, but you can appreciate how expression of many genes are different between these populations. And then we look at, we really wanted to trace these cells. We wanted to see how fast they come, how quickly they differentiate, and why they end up localizing in the perivascular areas. And those are, for us, from cancer biology perspective, are important regions, because those are enriched with so-called glioma stem cells or glioma stem-like cells that are the most resistant cancer population in these tumors, and are believed to give a rise to tumor recurrence. So to lineage trace these cells, we took advantage of multi-photon excitation microscopy that allows in a measuring of the cell dynamics, movement, and migration over the time. To do that, we generate with RCAS-TVA, we have the ability to generate tumors very shallow, superficially, and when tumors emerge, we take the cranial, we implant the cranial window there, and we do dextran that we can visualize vasculature, but it's also very easy to see because these tumors are pretty hemorrhagic. You can put the square in a peritumoral area to image microglia, and inside of the tumor to image bone marrow-derived cells. And this is just to show how we used CX3CR1 mice, only CX3CR1 GFP mice, because CCR2 RFP reporter is very weak, and with the differentiation, intensity goes down. So it was very inconclusive to use that reporter. So we had to go back and do bone marrow chimeras for imaging. This is just a naive coronal section of the brain, and staining is for CX3CR1, beautiful microglia uniformly distributed everywhere. And we wanted to image microglia movement over the time, and we did this for hours and days. As you can see, microglia cells, let me see why this, okay. Microglial cells, they extend and retract their processes, but they don't move. And when we looked at the monocytes, just to remind the cells, this is a reconstituted mouse, the green cells are monocytes. They are very small progenitor cells, and they're, they're supposed to be in the circulation moving around. So when we did the same, we needed to do the same experiment in tumor-bearing mice, we quickly realized we have 40% of tumor mass constituting of these cells. We are not going to be able to lineage trace these cells on single cell resolution level because of the abundance of the cell. So we had to go back and do bone marrow reconstitution experiments when we would dilute the bone marrow with the green bone marrow and black bone marrow. And in this case, we pretty much are labeling BMDMs or microglia. But what we're doing, we're diluting the color. So only 50%, for example, in this reconstituted mouse, cells that are going to be green. Only 50% of PMDMs are going to be green. Microglia is going to be black. The 50% of PMDM are going to be black. So we're diluting the cells to be able to image them. And we can do the same bone marrow chimera to image microglial cells. We reconstitute CX3CR1 GFP bone marrow with the black bone marrow. So all the cells that come from the bone marrow are going to be black, and microglia will be only the green cells. And this is how the reconstituted mouse looks like. And this is what we get when we have a tumor. So as you can see that microglia density is very low in the tumor, but you see microglia accumulated in the peritumoral areas, activated microglia. So we image the microglia in a tumor. Similarly, what we saw in a naive microglia, these cells extend and retract their processes, but they don't move. This is probably why they get pushed out in the peritumoral areas with very fast-growing tumor cells, and they form these reams of the cells in a perivascular, peritumoral areas. And based on what's known in literature and in our preliminary data, we have microglia in a peritumoral areas actually promotes the tumor invasion. And this is a bone marrow reconstituted mouse. So any green cells we see here will be bone marrow derived. And this is when there is no tumor. And this is when we have a tumor. So majority of cells within the tumor come from bone marrow. And when we lineage trace and look at these cells by imaging, you can see smaller the cells are, more progenitor staged when they exit the brain-blood barrier and get into the brain parenchyma, more dynamic they are. But when they get differentiated into larger size cells or become more macrophage-looking cells, they become more stationary, similar to microglia, like here in this corner. They extend, they retract their processes, but they don't move. This is why they end up, probably because the differentiation process is pretty fast, they end up being in a perivascular areas next where the most glioma resistant cancer cell population is located. So by using IMARIS, you can pretty much do what happens. It's true, there is a correlation, higher GFP intensity, less movement. So low GFP cells, which are the inflammatory newly infiltrated monocytes, they are highly dynamic and they move around. But when they differentiate into macrophages, they upregulate GFP expression and they stop moving. So it makes sense because macrophage is more stationary cell, while monocyte is more moving. And we can trace and do dynamic of the movements that we've done. And this is just checking the movement of microglia, which is really, the body doesn't move. So to understand how these cells come, whether that's the compromised brain-blood barrier or chemokine gradient, or both contribute to it, we started with the brain-blood barrier. So we use anti-VEGF antibody, which is one of the drugs that within 30 years we got approved for recurrent GBM. It's called bevacizumab, or anti-VEGF antibody. What it does to vasculature, it decreases the leakiness. And so we did the tumor implant, and then we start treating this mice with anti-VEGF antibody to normalize the vasculature. You can appreciate, you can see the difference in vasculature already. And then we did a cranial window in this mice, and we imaged the cells. So you can see there is significant decrease in monocyte infiltration. That's at, brain-blood barrier is compromised. But it's interesting, it's not just the physical hole that cells can come, because we have infiltration of most rarest population of myeloid cells in circulation. We have a lot of neutrophils that don't come, but we have less inflammatory monocytes, and those are the ones that come. So it means that it's true, the brain-blood barrier contributes to the infiltration for the cells, but it's not the only mechanism that drives the cell infiltration. So that's one. With this, we decided, okay, now we have to look at the chemokine gradients. We're going to look what tumor cells produce to bring these cells. And of course, the first choice was monocytes chemoattractant family proteins, and those are 4 members there. We refer to them in literature as MCP1, 2, 3, 5, and 4. It's different in human and mouse. Or we call them CCL2, 7, 8, 12, and 13. So this family is important for monocyte exit from bone marrow, and those chemokines, especially this 2 signal through CCR2 receptor, which is expressed in inflammatory monocytes. And so it's interesting that this chemokines, and they are localized right next to each other. It's important because it made our life difficult. We couldn't cross single knockout mice into each other to create a quantile knockout. So similarly in human, they are also localized next to each other. So that tells 2 things. First, we can't get a mouse that will lock all of them. And second, that there will be a redundancy in function in this chemokine, meaning targeting one, the other one will compensate. And that's exactly what we got. So we started with CCL2, which was the most studied one, which is important for directional movement of monocytes into inflammatory sites and exit from bone marrow. We stratified the cancer genome atlas, glioblastoma patients based on CCL2 expression. We divided them into CCL2 high, it's pretty much 25%, and then the bottom 25%, which is CCL2 low expressing. So I just want to highlight, and we see that CCL2 high expressing tumors have shorter survival time. Now it's just to say that tumor cells do not respond to CCL2. So any effect we see here comes from the effect of macrophages that we see. And then to mimic the same situation with high and low expression, we took advantage of CCL2 heterozygote mice, and we crossed this mice into our TDA transgenic mice. And by overexpressing of PDGFB as an oncogene and silencing p53 tumor suppressor, we created GBM in this mice. And it's true, heterozygote decreased the curves look like in human. But here we have overlap, and that tells there is a redundancy in function. So I won't bore you now with CCL7, 8, and 12 because we got exactly the same result. In each case, we get shift in the survival, and we get trend, but not significant decrease in the infiltration of these cells. Then, of course, we couldn't cross the mice because they are in the same chromosome. We had to be a bit more creative, and we took advantage of CRISPR-Cas9 editing to deplete all the MCP genes in the same chromosome. So we removed CCL27, CCL11, CCL12, and CCL8. And we validated the mouse. We just gave an LPS dose. And at 24 hours later, when we collected the serum, you can see that all the chemokines that we target by CRISPR-Cas are gone, and CCL5 we used as a positive control, it's there. And when we look in this mouse, and this is the t-SNP plot, we did this in collaboration with our macrophage experts in development, Elver Goysen at Emory, we saw that the biggest effect we see, it's the decrease of monocytes in the blood, and they get trapped in bone marrow, so there's no cell killing, so they can't exit the bone marrow. And this is the biggest we achieved so far compared, like CCL7 is just a reference of single chemokine, number of decrease in inflammatory monocytes in circulation. But cutting all 5 chemokines in human is difficult. We learned from this, though, that single chemokine targeted therapies will not work in GBM, and they haven't been successful. But we were looking at something upstream regulating these chemokines, and we came across to interleukin-1 beta, which is a potent pro-inflammatory chemokine. We looked, this is from Allen Brain Atlas, RNA sequencing from regions, and you can see IL-1 family members are in the perinecrotic, that's where we have macrophage-enriched areas, and in a perivascular region, which I said bone marrow-derived macrophages are localized. And when we look, there's a positive correlation between IL-1 beta and IL-1 alpha samples expression, which is not surprising, they regulate each other expression. And when we removed IDH1 mutant tumors from TCGA, because those are very different tumors, and we focused on IDH1 wild-type tumors, we stratified the patient with IL-1 high and low expressing. We saw very clearly that IL-1 expression correlates, high levels correlate with shorter survival. This is just IL-1 pathway, which is very complex. It was very interesting for us, particularly because IL-1 can be expressed with many cells, but only myeloid lineage cells, neutrophils and monocytes and macrophages can cleave IL-1 through inflammasome formation into biologically active form that can be released and do biological function. And when we look at the TCGH, we're like, look, expression of other IL-1 downstream targets, we saw that it's true, it holds true that from the beginning to the end, IL-1 pathway high expression correlates with shorter survival in GBM patients. So when we stained patient samples, GBM samples with IL-1 and IBA1, which is a pan-macrophage marker, labels macrophages, monocytes, and microglia, we can see that there is a nice overlap. The macrophages are the one that produce IL-1. Unfortunately, in mouse tissue, we couldn't stain because we had the knockout and all the IL-1 available antibodies really would stain the knockout. So that there's no good quality IL-1 antibodies that work in a mouse. But you can see this is the cytopalisading necrosis, and where we see the massive infiltration of these macrophages and in the perivascular areas. And when we did that, we sorted naive microglia, tumor microglia, BMDMs, and glioma cells. They all express, but the highest expression is in BMDMs, bone marrow-derived cells. And we know, although tumor cells express it, they can cleave IL-1. We never seen tumor cells to be able to cleave IL-1. And then this is just IL-1 ELISA from tumor samples to show that IL-1 levels are really high in the tumors. So then to understand, does the tumor induce IL-1 expression in myeloid cells? We do the culture experiments, we culture microglia and we culture macrophages from bone marrow, and then we did the co-culture experiments with brain slices. And the moment we cultured the slices with the cells, they induce both IL-1 alpha and IL-1 beta expression. So tumor cells induce IL-1 alpha and IL-1 beta expression in myeloid cells. But what IL-1 does to tumor cells themselves? So here is the cultures. We can culture these glioma cells in a primary cultures, and in early passages, they retain beautifully the morphology and behavior of tumors. And if we stimulate those with the IL-1 beta, you can see MCP family chemokines, all 4 of them are induced. And this is on RNA level. And then you can see by ELISA for CCL8, CCL7, 12, all proteins are induced too. So IL-1 comes from myeloid cells, signals to tumor cells, and induces monocytes chemoattractant protein expression by tumor cells. Now, tumor cells themselves, if we stimulate with these chemokines, they don't respond to them. So the only reason they upregulate these chemokines to recruit more macrophages into the tumor. To understand how, because IL-1 regulates, it's not transcription factor, but IL-1 is, we looked at the downstream targets of IL-1, and we saw beautiful induction of NF-kappa B pathway when we stimulate by IL-1 tumor cells. And NF-kappa-B is known that all these 4 chemokines have NF-kappa-B binding sites. So we think it's through NF-kappa-B activation that IL-1 induces MCP network in tumor cells. What does IL-1 to tumor cells themselves? So one of the measurements that we do for aggressive behavior of tumor, we measure the stem cell frequency, cancer stem cell frequency in these tumors. And we do that by measuring site population in a murine models. And the site population represents stem cell. It's a functional stem cell assay. And this is the population. And this population doesn't allow Hook's dye that we do to get into the cell, because stem cells have higher expression of ABCG transporters, and they pump out the Hook's dye. So the Hook's dye doesn't get there when main population picks up the dye. So if we culture our primary cells in FBS condition, which is not enriched for stem cells, we don't see difference here, which is not surprising. But when we culture them in a neural stem cell condition and retain their phenotype that we see in the perivascular areas, we can see that IL-1 stimulation doubles the stem cell frequency, and probably that's how it promotes tumor growth. And we do the same experiment, and unfortunately, site population assay is not working very well in human. There we use limited dilution assay when we dilute the cells and follow the colony formation from the single cell. So you can see that if we add IL-1 beta, stem cell frequency increases. And if we add IL-1 receptor antagonist, which won't allow IL-1 to work, we shift the frequency, we reverse the effect. So IL-1 really promotes tumor growth by increasing stem cell phenotype in tumor cells. If that's the case, if IL-1 really regulates through NF-kappa-B, this was all in a culture conditions, we wanted to see if that in vivo happens. To do that, we have the library of all IL-1 and inflammasome mice. So we generated tumors in IL-1 alpha beta double knockout, and we did double knockout to avoid the redundancy of alpha and beta. And when the tumors emerged, we took the tissue and did ELISA, and you can see all the chemokine levels are decreased. And here we used VEGF as a positive control, which shouldn't be affected by IL-1. So then we said, okay, now we got to generate tumors. And since we know that IL-1 is only can be processed into its active form by myeloid cells, and we did the experiments to show it doesn't happen in tumor cells, we transplanted wild-type tumors into IL-1 beta knockout and IL-1 beta wild-type mice. And this is a dramatic difference, and this difference is specifically driven by tumor microenvironment-derived IL-1. And when we look what happens to the tumors, do we see decreased myeloid infiltration? This is the total CD45, CD11B population. You can see significant decrease. And when we look down on whether this effect is on microglia, we don't see changes in microglia numbers. Here, microglia in a wild type and a knockout. And the biggest difference we see in a bone marrow-derived monocytes, macrophages, their number is significantly decreased. So does the number of LY6C high inflammatory monocytes is significantly decreased in IL-1 beta knockout mice. But those are germline knockout mice. We wanted to do experiments by treating pharmacologically, blocking IL-1, see if we have an effect. To do that, we generated tumors 2 weeks later, because IL-1 receptor antagonist and antibody will have a problem to get into the tumor. We wanted to do proof of principle experiment and make sure that the delivery of this antibody and anakinra, which is the receptor antagonist, is not compromised by brain-blood barrier permeability. We transplanted the cannula exactly into the bar hole that we do the transplant the tumors, and so directly into the tumor. And we mixed that with a low concentration of Evans blue that shouldn't have any effect, but pretty much allows us to see how much diffusion we get of the antibodies and antibody and anakinra. And you can see it's around roughly 60-70% diffusion rate. And when we look at the survival, so both anakinra, which blocks the receptor, and anti-IL-1 antibody direct delivery to the tumor extends the survival of tumor-bearing mice. And when we look whether macrophage number is decreased, this is the control, and this is IBA1 staining quantification. In both cases, we see that the macrophage number is decreased. So if I could summarize, we have an IL-1 beta IL-1 receptor protumorogenic loop in glioblastoma. What happens, we have monocytes that exit bone marrow in response to monocytes chemoattractant protein. They get into circulation. Our tumor cells increase MCP levels to attract monocytes from circulation to the tumor. So those guys come into the tumor, they produce IL-1 beta, and then they secrete that IL-1 beta, and tumor cells respond to IL-1 beta, but increase tumor progression and shorten survival of tumor-bearing mice through increased stemness there. But this is one of the mechanisms, and for sake of the time, and I'm okay, I'm going to be on time. This is just that Alice made, a new fellow in the lab, made an illustration comparing the macrophages to a Cerberus from Greek mythology guarding the underworld. And today I talk about macrophage function and tumor progression, how they induce, and IL-1 was an example. But we are also looking at how IL-1 modulates through macrophages immunosuppression. And we recently demonstrated that IL-1 secreted by myeloid cells can affect vascular permeability by affecting directly into endothelial cells. And this all was looking at the macrophage biology within the same tumor. Now, what happens? Do we finish it? No, it was the intratumor heterogeneity, and that was all our focus so far was. And there is one of the reasons so far we don't have a macrophage targeted therapy that successfully works in GBM, although we have 40% of cells being macrophages. It's the intertumoral heterogeneity in the cells. And I just want to give a few slides, and I will keep the data for the next time if I give a talk, to say that that was a PDGF-driven tumor. But now we have different driver mutation tumors. We have NF1 silenced tumor, EGFR ever-expressing tumors. And if you look at just the big picture immune landscape, you can see that those tumors are very different. Their immune microenvironment is very different. And their macrophage function is very different. So meaning that driver mutations create different macrophages. So we have a lot to go to understand how TAM compartment works and how driver mutations, for example, in adult GBM, affect heterogeneity. And we do need to understand that because in human, we have all these subtypes happening, and we have to develop macrophage therapies that will work in all the subtypes. And then when the picture comes even more complicated in pediatric brain tumors, that we are also very interested, because here we have tumors that form in a different locations in CNS. You have in a brainstem midline gliomas, and you have hemispheric. And if we did an RNA sequence, we did RNA-seq with the immune-related genes by nanostring, and we're now doing a single-cell sequencing. If you cluster them in human, if you do brainstem tumors and hemispheric, you can see that brainstem tumors, the IPGs or DMGs, they have a higher inflammatory score, and that high inflammatory score correlates with shorter survival time of the patient. And if we do the same thing with the same driver mutation, with the same location, and with the same histone mutation in a mouse models, and we cluster their immune thing, you can see that similar what we see in human in a murine DMGs, also inflammatory score is higher. And of course, with murine models, we can ask the questions whether it's a driver mutation or tumor location dictates that. And this is the combinations that we use to generate the pediatric brain tumors and study heterogeneity of TME. You can see that if we use the same oncogenic driver, which for example, PDGF, and we generate tumors wild type for histones in a hemisphere and versus the brainstem, you can see brainstem microenvironment is very different, meaning that's true, driver mutations matter, but also tumor location in CNS matters. And if we do the same brainstem gliomas, but in one case we use 3.1K27M mutation, oncohistone, and in the second case we do 3.3K27, we can see that 3.3K27 have a higher inflammatory score, and from all the pediatric high-grade gliomas, those are the ones that have the shortest survival time and more aggressive. So there is a lot more work to do to understand how different TAM compartment is based on tumor location in CNS, based on the driver mutations in pediatric, based on the oncohistone mutations. And and I just want to leave this with probably saying that it's essential for us to understand both inter and intratumoral heterogeneity of TAM compartment if we want to develop successful therapies aiming to eliminate the supportive niche provided by myeloid cells. And by this, I want to thank everyone, and I hope I didn't speed up. I have a tendency of speaking very fast, especially when I get excited about this stuff, and I do every time I do. But I'm happy to take any questions you have.
Fantastic, Dolores. Thank you so much. I really enjoyed that a lot. We do have time for questions. So I'm going to ask if you could please unshare the screen, and that way we could see one another again. And I'll just ask people who want to have questions for you to unmute themselves and ask away. Praveen, go ahead.
Dolores, that was amazing. Really beautiful work. So the question I have for you is, post-treatment, is there any data that the myeloid compartment is different or changes?
Yes, yes, there are the data. First of all, I mean, I promise my lab I won't speak about their new data, but I'll give a little bit. Yes, myeloid compartment changes. So, you know, post-treatment, for example, in adult tumors, right, treatment usually induces radiation therapy or anti-VEGF therapy, induces the transition of these tumors into more mesenchymal looking, right? And that's in a mesenchymal tumors, we have less monocyte infiltration, more neutrophil infiltration. So if we take and treat these tumors, the ratio of neutrophil and monocytes in recurrent tumor changes. And the preliminary data we have, we use CXC, CR2 in antagonists to block neutrophil infiltration in the IPGs. There we see with radiation, massive infiltration of neutrophils. These two are very tight. And you manipulate one, you will induce infiltration of another one, which is interesting. What if we block both? What's going to happen, right? $1 million question. Can we get T cells into the tumor?
Well, it makes you wonder then, when anti-macrophage therapies have failed in humans, most of those are treated in the recurrent setting. I think very rarely are they ever at the primary.
That's a very good point. So that brings back into CSF1 receptor inhibitor, which is very efficient of depleting naive microglia in the brain, but has been not very successful. It worked beautifully in original papers in PDGF-driven tumors. And you're right, the recurrence is more mesenchymal. We don't see efficacy with CSF1 receptor in mesenchymal tumors. So it's not surprising that it didn't work in recurrent. That brings back to my slide. I think we need to test these therapies in multiple models with different driver mutations that are common in human disease, because then we take it into recurrent, and we do the trial on unselected patients, right? With one model. So yeah.
Thank you. No, that was great.
That's very true.
Other questions? Dolores, I was struck by one of the latter slides you showed of the different driver mutations, PDGF and EGF and so on, resulting in very different immune-like characteristics on your RNA sequencing data. Do you know what it is about those driver mutations that are eliciting such distinct immune environments of the tumors?
It's a very good question, Eric. It's just the question I think it's essential to understand. So that's tumor cell that has a EGFR V3 mutation, different from the tumor cell that you induce PDGFB. Yes, they have different signaling downstream. They have a lot in common too. Or NF1 loss activates different pathways, right? And that changes the signaling signature of tumor cell itself. And then obviously that will impact also what chemokines are secreted by tumor cells. And obviously that will determine the ratio, which activation, because the cells, myeloid cells, are very plastic cells. They are genetically stable, but they are very quick to change their expression profile. And depending what signals they get from tumor cell, their expression profile will be different. Now, NF-kappa-B pathway, for example, is a classic example. So PDGF-driven tumors do not have a constitutively active NF-kappa-B pathway, but NF1 deletion leads to RAS activation, and this tumor have constitutively active NF-kappa-B pathway. means the chemokine levels are 10 times higher in these tumors compared to the PDGF-driven ones.
I
Do
see.
you see more macrophages there? You see twice more macrophages in mesenchymal tumors if you look.
So you see it more as the factors that the tumors are secreting as opposed to cell surface proteins on the cells of the tumors.
Correct.
Yeah, interesting. Very cool. So Marina Varghese has a question. Marina, if you're still with us, if you want to unmute yourself and ask the question yourself, otherwise I could read it.
Thanks, Eric. I can ask.
Oh, excellent, Marina. Yeah, go ahead. Go ahead. Better for you to ask directly.
I might get interrupted with some construction noise.
That's okay. Go ahead.
I was wondering, beautiful work, Dolores. Thanks for presenting. And I was wondering whether there's any differences you noticed functionally between the microglia and the infiltrating macrophages in the tumors. And as a second part of that question, do infiltrating macrophages also contribute to other disorders with leaky blood-brain barrier?
Yeah, it's a very good questions, both of them. I'll try to answer to the best of my ability. So now, microglia and bone marrow-derived macrophages. So their expression profile looks different. They have a common genes, which is not surprising, but the expression profile also looks different. Now, the location in the tumors also is different. So microglia is in a peritumoral area where we have invasive edge of the tumor, and the macrophages, bone marrow derived, are inside of the tumor in a perivascular area. So the microenvironment is a bit different in an invasive edge versus the perivascular areas. So based on that, we didn't do, of course, we don't have the ability so far as a field. We weren't able to specifically deplete microglia, leave BMDMs off, or do otherwise experiment. We so far, this MCP mouse is the only mouse we are able to almost nearly 200% block this cell infiltration, right? Before we didn't have the tools. This is the way you do it to be able to say the function in this is different from this, right? Without killing and calling in systemic inflammation that you don't know what the consequences are. But if I go back to the original work in organotypic slice cultures, Helmut Kettenmann's group for years been working on it. In organotypic slices from naive brains, where you have only microglia, you can ex vivo generate brain tumors there. And the effect that they saw when with cladronate, they depleted microglia in the cultures, they saw that tumor invasion is affected, not the growth, but the invasion. So fair to say, in our models, and also in a human, they're also localized in peritumoral areas. Fair to say, probably, and I'm speculating because I don't have the data, that microglia probably promotes tumor invasion in invasive edge because they produce MMPs, and they can degrade the ECM there and facilitate tumor cell invasion. And you have a beautiful two groups there that show that in your department, Jenny Zhao and Roland. And their work is mainly focused on microglia promoting corralling, facilitating and tumor cell invasion. For us, the focus always been more on a myeloid from blood circulation coming there. And so in other CNS diseases, I mean, MS is probably one that is closer to GBM behavior. And I actually got inspired by Richard Ransahov's work on MS using this red and green mice. We didn't get clean result on only red and only green that they saw in MS model. We see mix. But there, their function is very different. We had a lab next to us on working on a viral infection in CNS, Connie Bergman. In there, they saw massive infiltration of these cells within 7 days of infection, but then these macrophages disappear after 10 days, they are not there, they're gone. We don't see that in GBM. They come, they form a like symbiotic network with tumor cells, and tumors, from the moment tumors emerge in mice and kill the mouse, it's 2-3 weeks the most. So you don't really have time to trace the first myeloid cells that comes and differentiates and how long it lives. So, but it seems to me their life is extended in tumor context, which is full of this chemokines, growth factors, anything you need for survival for them. And so their function can be very different from disease context. Do I believe they have different function from microglia? Yes, but I think we still don't have the ability to eliminate these cells really for function. And what's going to happen if we block? Are the other cells are going to come, which probably what's going to happen. Hope that answers.
Excellent.
Yes, thank you.
Yeah, excellent. So any last questions? We have a couple minutes left. Okay, with that, Dolores, thank you again very much. I was impressed by the application of just state-of-the-art molecular tools to understand the heterogeneity of brain tumors and their molecular and cellular mediators. Really beautiful work.
Thank you.
So thank you so much for joining us. And thank you, everybody. And enjoy the rest of the week. Bye-bye.
Thank you very much. And bye-bye, everyone.